Secondary metabolites are diverse organic compounds produced by plants that aren't directly involved in their growth, development, or reproduction. Instead, these compounds often serve crucial ecological roles, such as defending against herbivores, competing with other plants, or attracting pollinators. Although secondary metabolites are not necessary for the plant's immediate survival, they are vital for the plant's long-term fitness and interaction with its environment. Secondary metabolites are categorized into several groups. Alkalides, nitrogen-containing compounds that often have significant pharmacological effects. Examples include morphine and quinine. Terpenoids, the largest class of secondary metabolites, including essential oils and plant resins like menthol and carotenoids. Phenolics, compounds with aromatic rings and hydroxyl groups that contribute to the plant's color, flavor, and defense mechanisms. Common examples are flavonoids and tannins. Glycosides, these are compounds where sugars are bound to other functional groups via glycosidic bonds, such as saponins and cardiac glycosides. Plant tissue culture is a powerful technique used to grow plant cells, tissues, or organs in a controlled, sterile environment on a nutrient medium. This method allows for the efficient production of secondary metabolites, offering a sustainable alternative to traditional plant harvesting. By using tissue culture, we can produce these valuable compounds in a controlled environment, ensuring consistent quality and supply, regardless of seasonal or geographical variations. The process of producing secondary metabolites through plant tissue culture involves several key steps. 1. Selection of plant material. The right plant species and tissue must be chosen based on the desired secondary metabolites. For example, Catharanthus roseus is selected for alkaloid production, specifically vincristine. 2. Establishment of aseptic culture. The plant material is sterilized to prevent microbial contamination. This is achieved through surface sterilization using agents like ethanol or sodium hypochlorite. 3. Initiation of callus culture. Here, undifferentiated cell masses, known as callus, are formed from explants. This is done by placing the explants on a solid medium containing a balanced ratio of auxins and cytokinins. 4. Optimization of culture conditions. Environmental factors such as light, temperature, pH, and medium composition are fine-tuned to enhance metabolite production. For instance, some metabolites require dark conditions for optimal production. 5. Cell suspension culture. This step involves using a liquid medium to maintain and proliferate plant cells in suspension, ensuring even distribution of nutrients and oxygen. 6. Elicitation. Elicitors, which can be biotic, like fungal cell wall extracts, or abiotic, such as UV light, are used to stimulate the production of secondary metabolites. For example, jasmonic acid can increase taxol production in Taxus species. 7. Precursor feeding. By providing precursor molecules, the biosynthetic pathway is enhanced. For instance, feeding phenylalanine can boost flavonoid production. 8. Scale-up and bioreactor cultures. Transitioning from small-scale cultures to large bioreactors allows for industrial production. Different types of bioreactors, like stirred tank and airlift, are used depending on the metabolite. 9. Extraction and purification. The desired secondary metabolites are then isolated and purified using methods such as solvent extraction, distillation, and chromatography. 10. Analysis and characterization. Finally, the identity and purity of the secondary metabolites are confirmed using spectroscopic methods, mass spectrometry, and chromatographic techniques. The secondary metabolites produced through plant tissue culture have wide-ranging applications. Pharmaceuticals. They are used to produce medicinal compounds such as vincristine, artemisinin, and paclitaxel. Agriculture. Natural pesticides and herbicides developed from secondary metabolites reduce the need for synthetic chemicals. Cosmetics. Natural fragrances and pigments derived from these compounds are valuable in cosmetic formulations. Research. Plant tissue culture also facilitates the study of plant metabolic pathways and the effects of various stress factors on metabolite production. Despite its many advantages, plant tissue culture presents challenges such as high initial costs and the need for extensive research to optimize conditions for each plant and metabolite. Additionally, skilled personnel are essential to manage and operate tissue culture systems effectively. However, the potential of this technique for sustainable and scalable production of secondary metabolites makes it a valuable tool in various industries.